products, reactants, energy, and enzymes. So let's talk about chemical reactions. It's a process that changes or transforms one set of chemicals into another. Mass and energy stay the same during chemical transformations. Now, some reactions are slow, like iron and oxygen forming rust, while others happen pretty rapidly. Elements or compounds that enter a reaction are called reactants. The elements or compounds produced by the chemical reaction are called products. Chemical reactions involve changes in the chemical bonds that join atoms and compounds. For example, an important chemical reaction in your body is carbon dioxide being removed from your blood. Well, what does all this mean? What are these reactants and products? Well, here you've probably seen something like this, a chemical formula. So on the left side of the arrow, which means yields, we have the reactants. They combine together, they react together, and they form products. So, for example, when we talked about iron and rust, you can take, or iron and oxygen, you can take iron and oxygen, they react together and form rust. Here in this example, we have 2H2 plus O2, and you're going to get the product 2H2O. So reactants always on the left side of the yield arrow, products always on the right side of the yield arrow. Now, chemical reactions can release or absorb energy. The change in energy determines whether or not a reaction will occur. Chemical reactions releasing energy occur on their own, while those that absorb energy will not occur without a source of energy. They're not going to occur on their own. Energy can be released as heat, and in the case of an explosion, for example, light and sound would be the energy being released. Now, organisms need to use energy in order to stay alive, so organisms must have a source of energy to carry out those chemical reactions. Now, plants gather energy from the sun, while animals, we eat plants or other animals to get energy. Now, humans release energy needed to grow, breathe, think, and even dream at night through chemical reactions done when we break down our food for energy. So when we're eating this delicious food here in America, we're not just doing it for kicks. We're doing it so we can grow, so we can breathe, so we can think, and even dream, and do all these other things that we need to do. And the food's going to be used to produce these chemical reactions that we need in order to survive. Now, activation energy. Chemical reactions that release energy don't always do it on their own. Now, that's great news because it means your paper might suddenly burst into flames. So the paper around you, if it wasn't for the energy, the activation energy needed, it would just suddenly burst into flames. Well, cellulose burns in the presence of oxygen. Cellulose is in paper, but it won't start without activation energy. Energy that is needed to get a reaction started. So how do we get cellulose to burn? What is the activation energy? Simple, a match. The match would be the activation energy in this example. Now, activation energy is involved in chemical reactions regardless whether the chemical reaction is going to release or absorb energy. Now, chemical reactions that make life possible are either too slow or need activation energy too high for them to happen. These chemical reactions are made possible by what is called a catalyst, a substance that speeds up the rate of a chemical reaction. Well, the reason catalysts work are because they lower a reaction's activation energy. In other words, they lower the activation energy required to begin the chemical reaction. Catalysts are found in nature, and we call these catalysts enzymes, which are proteins that act as a biological catalyst. Enzymes are extremely important and allow life to exist. So in your blood, carbon dioxide reacts with water to produce carbonic acid. Now, carbon dioxide would build up faster than your body being able to remove it. Well, that would lead to death. Well, an enzyme called carbonic anhydrase increases the reaction, moving that carbon dioxide out of your blood by 10 million, allowing it to move so much faster. And the enzyme names usually have something to do with what it does. In the case of carbonic anhydrase, all right, it increases the reaction. It's going to move that carbon dioxide out of your cells by a factor of 10 million, allowing you to survive. All right, the enzyme-substrate complex. This is something that is complex and a little difficult to understand, but we have an illustration to help you out. Now, in order for enzymes to work, there must be enough energy for the reactants to collide with, so existing bonds can break and new bonds can form. Now, enzymes provide a site for reactants where they can be brought together to react. Now, the reactants of enzyme catalyst reactions are called substrates. Substrates bind to a site on the enzyme called the active site. The active site and substrate have a very specific shape so that one fits in the other precisely, just like a lock and a key. Now, enzymes control pathways in a cell, making materials that cells need, releasing energy, and transforming information. What exactly does all that mean? So here we have a bunch of specialized words, a lot of vocab, and a lot of complex information, and you can read this as many times as you want, and you're probably not really going to know what it means. So let's go to an illustration. So here we have a bond. This is a substrate. This is sucrose, which is glucose and fructose bonded together. So here's our enzyme. Here's our substrate. All right, it's going to bind together like a lock and a key. Once it binds together, we have what's called an enzyme-substrate complex. So here we have the substrate. Here's our enzyme. They come together. They fit together like a lock and a key. And once they fit together like a lock and a key, you're going to get the enzyme-substrate complex. Now, the binding of the substrate and enzyme places stress on the glucose-fructose bond. So where it has a bond, it's bound together. It's placing stress on that bond, and it's going to break it apart. So here the bond has been broken, and then the products are going to be released. So the reactants come down as substrates, fit together like a lock and a key, and when it binds together, it produces stress on that bond, breaking the bond apart, and you have the products that can be released. So here comes glucose, free to go wherever it needs to go. Here comes fructose, free to go wherever it needs to go. The enzyme goes on looking for a new substrate. So everything that we just looked at, that's what it means. That's how it works. So now that we know what all this means and what it looks like, we can wrap it up. So enzymes can be affected by temperature, pH, and regulatory molecules. Now, regulatory molecules carry chemical signals inside cells that switch enzymes on or off. That's their job. If the enzyme needs to be used, it'll turn it on. If it doesn't need to be used, the regulatory molecules will switch it off. Temperature also affects enzymes, and most enzymes work best at 98.6 degrees. Well, isn't it our lucky day, because that is our body temperature. Now, pepsin, a stomach enzyme which starts protein digestion, works best in acidic conditions, which is what your stomach has. Your stomach has a very acidic environment. So this is an example of pH affecting enzymes. Now, pepsin wouldn't work well outside an acidic environment. So when we talk about temperature, pH, and regulatory molecules, this pepsin is an example of pH. If you have any questions, you can always post a comment below or give us an email, and we'll see you guys next time.